So now we're going to start talking about the different types of macromolecules that are out there. And there's going to be four basic types: proteins, nucleic acids, lipids, and carbohydrates. And with the exception of lipids, they're all going to kind of follow this format where they're going to have that same building blocks that we've been talking about to form those polymers. So the first one we're going to talk about are going to be proteins. And proteins are going to be crazy molecules, a lot of variation in their structure, a lot of variation in their function. And so I've listed here a bunch of functions that they do: enzymes, defense, transport, support, motion, regulation, and storage. So lots of different things that proteins can do. Basically, when in doubt, a protein does it. They do tons of things. If you look at this, it kind of gives you an idea too. Proteins make up more than 50% of our body's dry weight. So that means if we sucked all the water out of our bodies, half of the remaining weight would be proteins. So I'm not lying when I say they do a lot. So as far as their structure goes, they're going to have amino acids being their monomers. So what I mean is, if we have our setup here of a polymer, these little circles are going to be amino acids. Okay, so they are going to be the building blocks of proteins. So if we are going to look at a close-up of a molecule of an amino acid, it's going to look like what you see right here. So a carbon, and then it's got an amino group coming off, it's got a carboxyl group coming off, a hydrogen, and then this one you may not recognize, the letter R. The letter R means that could change out for whatever amino acid we're talking about. So there's 20 of them, so the difference between all 20 of them is that R group. And so we're going to play a little bit more with this with the models in lab so you can see what we're talking about. Okay. Now the other thing is that all of these amino acids are going to be joined by a covalent bond, but of course we have to have a special name for that covalent bond, and that name is a peptide bond. So this bond right here, peptide bond. So every bond joining amino acids is going to be called a peptide bond. Okay, so I could say, what kind of macromolecule has peptide bonds joining its monomers? You'd say a protein, okay, or a polypeptide. Okay, so I brought up the next point, which is a tough one for people to understand. So a polypeptide is what we would call this right here. It's just a long strand of amino acids. It doesn't become a protein until it actually gets folded into its three-dimensional shape. And this is what I have written here. So to be clear, amino acid, peptide bond, amino acid, peptide bond, and so on, that's just a polypeptide. Then when we take that polypeptide and actually fold it in its three-dimensional shape, then it becomes a protein. So I like to think of a polypeptide like a string of yarn, and then if you knit it into a sweater, that's the protein. Okay? So actually, we'll come to that in a second. There's a couple of types of amino acids. You've got nonpolar and polar ones. You have ones that have a positive charge and a negative charge. The aromatics, which are going to have rings, and then special function ones. I'm not going to have you know these by heart or anything, but I just wanted you to see the different R groups that you can have. Now, this is going to come up in lab when we play with the protein models. All right. So when we look at a protein, that jumbled up ball of yarn, right, we want to look at it at different levels. So what we divide it into is what's called primary, secondary, tertiary, and quaternary structure. So I like to think of it as zooming in like you do on Google Maps, or zooming out. So if you're as zoomed in as possible on a protein, you're going to be looking at primary structure, the really, really, really detailed stuff. If you're looking way, way out really far, then you're looking at quaternary structure. Okay, so it's all the same protein, it's just how detailed of a view you're getting. Now if we go to, let's see, yeah, this will work right here. Okay, so quaternary structure is if we're way, way zoomed out, right, and that's what's over here. Primary structure is over here, and that's really zoomed in, okay? So we'll start with primary. Primary, by definition, is just going to be the sequence of amino acids that make that protein. That makes sense, right? That's as zoomed in as we can get on a protein. All right. If we were going to zoom out just a little bit on that, we're going to come across secondary structure. And secondary structure can come in two forms. It's either going to look like these coils, which is called an alpha helix, or it can be folded into what are called beta-pleated sheets. So here you can see a little snippet of those right here, okay? So secondary structure, zoomed out a little bit, you can see the amino acids right here, but what you're seeing even more is that shape that they're folding in. And we're going to talk about what causes that to happen in lab. Okay, now if we look at tertiary structure, that's this whole thing right here. And that's going to be the actual three-dimensional shape of the protein all folded up. You see how it's all folded up on itself inside there? So that's going to be tertiary structure. We can still see our secondary structures here. There's an alpha helix, there's some beta-pleated sheets everywhere, but we can't see that primary structure because we're a little too zoomed out. Then quaternary structure is going to be one or two or more tertiary structures linked together. Okay, so you can see one here, and then it looks like there's one back there. So those are going to be the different levels of protein structure, and it just depends on what type of aspect of a protein you're looking at, what level of zooming in that you're going to do. Okay, back to the notes. Now, we've talked about how a protein is folded. Very, very, very important in its function. And it's going to fold due to a lot of different forces. There's going to be ionic bonding, there's going to be hydrophobic forces, hydrophilic forces. But another thing that's going to be working are going to be chaperone proteins. They're called chaperonins. And as a rule of thumb, if you ever see anything ending in INS, that's usually going to be a protein most of the time. Okay, so if we look over here, this is a good picture kind of showing you how a chaperone protein works. So what happens is you have your polypeptide. Notice it's not folded in its 3D shape, right? So that's why we call it a polypeptide. It goes into a chaperone protein, and the chaperone protein is going to help it to fold in the way that it should, and now we've got our folded protein. So that's the point of a chaperone protein. What's crazy is that there's diseases out there called prions that pretend to be these, and they misfold your proteins. And that's like what causes mad cow disease and that kind of stuff. Okay, so chaperone protein's really important. And going along with that, the folded up nature of your protein, extremely important. If it unfolds, then it's not going to function correctly. And so when it unfolds, we call that denaturation. Most of the time, a denatured protein cannot be refolded back. Okay, so let's talk about what types of things would cause that to happen. pH and salts can do that, and the way that they can do that is like this. So if that's your protein, very artistic. One thing that might be holding this protein together is ionic forces like that. Pluses and minuses attracted to each other, like that, and like that. So what's going to happen is if you change the pH, you're either increasing the amount of pluses around or decreasing them, right? That's what pH basically is. So if we were going to have another bunch of pluses here, all of a sudden this minus might be more attracted to this plus and might go in this direction, and now the protein's going to start to unfold. So that's why pH is really, really bad. Like a change in pH too much can cause problems with proteins. Another thing is going to be temperature, right? If you think about temperature, when you heat something up, the molecules move faster. So proteins don't like to be heated up, and if they get too hot, then they're going to just completely denature. That's one of the reasons why when a fever happens, we really monitor it, because if it gets too hot in your body, you're going to start having your brain proteins denature, and obviously brain proteins are kind of important, so you don't want that to happen. So you can kind of put this in real life ideas. If you think about a jellyfish sting, right? What do they tell people to do when they get stung by a jellyfish? They say pee on it, right? Well, your urine is going to have a different pH most likely, and so that hopefully will help to neutralize it. They also say put vinegar on it, and vinegar has a different pH as well. So hopefully that would help to denature those proteins that are in the toxin causing you to feel uncomfortable. As far as temperature, you know, when you cook an egg, right? You've got that clear egg white, and then when you cook it, it actually turns white. What you're doing and what's making it turn white is you're actually denaturing those proteins, and that's what's making it look like that. Same thing with a steak. You've got a steak that's nice and red. Those proteins are all nice and folded. When you cook the steak, it turns kind of brown or grayish, and that's the proteins denaturing. See all these fun things that you can talk about during dinner now or breakfast? I mean, it just never ends. Okay, now the next part we're going to talk about are nucleic acids. And nucleic acids are going to be functioning to store and transmit our hereditary information. So they're going to come in the form of DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. So DNA is basically our blueprints, and that gets passed on from one generation to the next. And RNA is a copy of the DNA that we use to actually make you into something physical. Don't worry, once again, we have an entire entire chapter that we'll spend on that. Okay, so remember with our protein how we had amino acids being the building blocks. Well, for nucleic acids, it's going to have the same exact setup, except the building blocks are going to be called nucleotides. So the same setup, but now the building blocks are nucleotides. So I won't have room to actually write this, but in between the nucleotides, we're going to have what's called a phosphodiester bond. So that's just the special name for the covalent bond between nucleotides to form nucleic acids. Okay? So I know that's a fun word, and don't worry, we will understand that a little later in the semester, why it's called that. All right, so if we're going to look at a nucleotide, here's one right here. You're going to have three parts to it. A phosphate group, then you're going to have a 5-carbon sugar, that's why it's called pentose, pent means 5, and then you're going to have a nitrogenous base coming off of it. So you can see here, here's one, here's another, and they can form big long chains. Okay, and in between them is going to be that phosphodiester bond. Now the nitrogenous bases, this orange stuff that you see here, you can see there's a little variation. Here these are single rings, and then here you can see double. And so those are going to be the difference between what are called pyrimidines and purines. Purines are going to have 2 rings, and pyrimidines are going to have 1. And so that's the only way that nucleotides are going to vary from one to the other, is whatever nitrogenous base they have hanging off of them. So remember in the amino acids, the way that they varied was what R group was coming off of them. So these guys are going to vary based on their nitrogenous bases. All right. So there's a couple differences between DNA and RNA. One difference is that RNA has only uracil instead of thymine. DNA has thymine, RNA has uracil. Neither of them have both of those. So that's kind of specific to each. The other one is that 5-carbon sugar. In RNA, it's called ribose, and in DNA, it's called deoxyribose. So that's just a difference there. So another thing about word endings is if you see anything end in OSE, that's usually going to be a sugar, right? If you think of sucrose and fructose and all those types of sugars we ingest. Okay, so that's going to be the differences between DNA and RNA. And then the last one is that RNA is just a single chain, and DNA are 2 wound together. So if I was going to draw RNA, it looks like that. But DNA is going to look like that, where it's got the 2 together. So that's going to be the difference between RNA and DNA. Now there's going to be a couple of special nucleotides out there that are going to be very important in our systems. One of them is going to be called ATP, and the other is called GTP. ATP stands for adenosine triphosphate, and that's a form of energy currency in our cell. Most of the time, our energy is going to be in that form, and we'll get into that in like chapter 8, chapter 9. GTP is another form of energy, but it's not as common as ATP. All right, in the next part, we'll finish up with the last two macromolecules, and then you'll be done with this chapter.